Morning, everybody. Just give people a minute or so to roll in, and then we'll get moving. All right, let's get cracking. Okay, so that's a little bit too far away. Closer. There we go. What do we need to do today? Oh, that's my reminder to teach genetics. Let's turn that off. All right, so today's all one word. What do we got? We need to finish up dihybrid cross, because we started the F2, we didn't finish it, so we're going to do that. And then we're going to talk about test cross. So this is a way of figuring out the genotype of a dominant individual, right? And then that's kind of it for Mendel. I wish I could draw like a natty little drawing of Austrian monk, but I can't. Now we're into probability and stats, right? So don't want to spend a huge amount of time on this because we've got a lot of this to cover, right? So this is going to be a wrap up. Then we need to spend most of the class on this. Move these out of the way. So what do we need to talk about? Probability. And so in terms of probability, we've got two different types. Independent, and or exclusive events. Hopefully you can see the writing there. Gets harder and harder to write clearly the further over there that I get. We should definitely be able to cover that today. Then let's see, hopefully we'll have time to get into scientific method, which is, you know, central to how we do science, right? And it's usually a mark of a sloppy scientist that doesn't follow this. And then hopefully we'll get into chi-square test, right? Which will be a essentially a recap, repeat, whatever word you want to use, of what we've covered in lab, right? So some semesters this comes first, other semesters this lab comes first, but either way you're going to have a second crack at the whip with this, right? So that's what we're going to aim to cover today. we should get at least down here. We'll see if we get, you know, all the way down to the bottom. Oh, hang on, let me get off of this. There we go. All right, so dihybrid F2 generation. So I'm very quickly going to write up what we were working on Tuesday. And then I'm going to fill out that table. I'm going to look at the ratios, and we're going to move on. So hopefully you've done that yourselves already, right? I can't stress enough that for things like dihybrid cross, chi-square, linkage problems, right, which will be in the second section of the course, the key to being good at them is to practice. Practice, practice, practice over and over again, and you will be able to do them almost without thinking, right? And so when you're in the exam, right, each question is equally weighted and they come up in random order and you have to do one at a time, right? So you can't come back to do a previous question that you skipped over, right? You have to answer the question and move on. So being able to do something like this, dihybrid or chi-squared or whichever, without having to go back through your notes and figure out how it's done, will save you a ton of time. And that time can then be spent pondering other difficult questions, right? So think about that. It's going to be like there is a reasonable amount of time for the exam, but there's not a lot of time for the exam, right? So being able to do these calculations and the like quickly and efficiently is going to be a big help. So practice, right? Don't leave this until the exam because you'll regret it, I think. Okay, so we have up here, we have our F1, which is, I do believe I used these letters, right? Black short hair. Same down here. Right, so those are our two F1 individuals. When's our first exam? That's a good question. I think it's in, actually, I'm not going to guess because I don't want to get it wrong. It is on the 16th of February for you lot. And we have a little bit of wiggle time to finish everything up before then because I've got a recap. Let me show you the, where are you going to see?
There was a question in the group chat yesterday. And the syllabus, it says they're online. Is that correct?
That is 100% correct.
Okay.
So you won't see them online right now because they magically appear on the morning of the exam. But they are online for the whole day of the exam.
Okay, thank you.
Yeah, you got a lot of flexibility when you take it. Still get the same amount of time, but you can start the exam whenever best suits you. Also, so there's the exam there in that one. You'll have an hour and 15 minutes, same as the class time. What else was I going to say? Oh, yes. Also, before I forget, Maria, I'm just going to get to your question in a second. I'm going to be on Zoom, this same link, on exam days during class time. So you don't have to come into class. I should answer your question, Marie. I mean, if you want to sit there and take your exam, that's fine. You have like patchy internet at home or something. But I'll be present on Zoom if you have any questions about the exam, accessing it, interpreting questions, stuff like that, I'll be in Zoom as if I were sitting in the class, you know, that kind of thing. But you don't have to take the exam during class time, right? If you wish to, that's great. If you don't, you could even use that period as a last gasp chance for asking questions. Obviously, they're not going to be questions that I'm going to help you answer on the exam, because you know, that wouldn't be kosher. But I'll be there to help you if you need it. I'll also be around and about the rest of the day. I'll try and check my email fairly frequently, you know, so that I can catch any questions or issues before they become a pressing problem. But I'm not going to be like able to guarantee to answer immediately, right? Because I might be out walking the dogs or working in the garage or something like that, or having my lunch. Okay, does that answer questions? We can talk about it a little bit more closer to the time as well. Awesome. All right. Let's get it moving. Oh, I saw a little thumbs up from Victoria. That's so cute. Thank you, Victoria. It's kind of like made my day, that. Anyway, where are we at? Yes, dihybrid. So, as always, come on, get your brain back in gear. Gametes. Same again for the others. The other side of the cross, essentially. I did actually make a mistake yesterday. I'm going to try not to do that today. I blame it on like weird angle of looking at this. That's my excuse. All right, there we go. So stick these together, then we'll work out the genotypes. I know for some of you, this is like, oh God, this is so straightforward. Some of you, this won't be the case. So that's why I'm doing the same for all of you. So I'm just going to rip through these. And you've got to look at what the cell kind of components are going to be, right? Can't just guess, which is what I did yesterday. Got it wrong, which was a bit embarrassing. That's that. All right. Sorry for the kind of wonky writing. It's a little hard to write these sometimes at an angle. Oh, my cricket's curled up like a little shrimp on the couch next to me. To show you that in a second. All right, there we go. So now she's unshrimped herself. Now what you do, right? So these are all the genotypes. We don't actually care a huge amount about the genotypes. What we care about is the phenotypes, right? The expression of those genotypes. And so now as we go through, yeah, I did that one right. As we go through, what you have to do is see whether or not there is a dominant allele for either of the genes, right? And so first we're going to look for the presence of a dominant allele for both genes. That will give us the dominant parent phenotype, double dominant, right? So that's this one, that one, that one. Not that one, which was a mistake I made yesterday. These ones. Oh, that's a squish cat. I think that's it. So, for those individuals, so the double dominants, yeah, I guess that's right. There should be 1, 2, 3, 4, 5, 6, 7, 8, 9. Right, so that's the dominant parent, right? How many, and I often ask questions on these kinds of things. How many individuals with the same double recessive phenotype combination are there in this as the recessive parent? Look again, everybody. How many in here are double recessive? Right, so you've got to have a double lowercase b. Yeah, everybody's getting it now. And a double lowercase l. Right, so we've got a double lowercase l here, but we've got an uppercase b. We've got a double lowercase b here, got a dominant l. That's right. The one you're looking for is this one down here. This is your only double homozygous recessive, right? So this one, what was it? Purple? Long hair. There you go. That's a purple long-haired cat. Right. So the number of individuals that you would expect to have the same phenotype combinations, I'll drop that down a little bit, as the P0 generation, the original parents, is going to be 10 out of 16. You're going to have 9 double dominant, you're going to have 1 homozygous recessive or double recessive, right? So if we were to take 10 out of 16 and simplify it further, that's 5/8. That might just be an answer to a question in the exam. I'll have to check. Okay, so essentially, a bit over half of the total possible outcomes are going to be the same as the parents. The rest are going to be a different combination of phenotypes, right? Phenotype combinations that you don't see in the parents. So let's find the one which is purple and smooth. Oh, there we go. Why don't I ask you lot? Which ones of these remaining cats are going to be not purple and smooth? Again, my corn and cats mixed up. Purple short-haired cats. Which one was that? That was, so this one over here, Haley, that is most definitely purple short hair. And Joshua, you're right, there are 2 others, 3 in total. This one here, again, what you're looking for is that. Only the individuals that are homozygous recessive for, yeah, that's right. Thank you, Joshua. Homozygous recessive for the coat color gene will be purple. But they also have a dominant allele for fur length, so they're short fur, right? So that gets you three of those. And then the final one is now you're going to be looking for homozygous recessive for fur length. And that's that one. It's actually kind of easy because they're the only ones left. But nonetheless, those ones. Right, so these are going to be black cats, but they're long-furred cats because they're homozygous recessive. Don't know why I bother drawing ears on them. You can't see it once I do this. They look like the coal puffs out of Spirited Away. I don't know if anyone's seen that film. It's really, really cool. There you go. So that's what gets you the 9:3 to 3:1 ratio. And these rather, are new combinations that you don't see in the parents. Yeah, I love that film too, Alfred. It's got to be one of my favorite of that style. Obviously, Howl's Moving Castle is also super, super cool. I like that one a lot as well. But actually, it's funny. My youngest daughter, for a while, she used to have a really squeaky sneeze, and it sounded just like the little hamster in Spirited Away, or mouse, or whatever it was. I can't remember. Okay, so that's that. Any questions before we move on? Give me a little sleeping pup pic. It is truly the life of a dog. Cricket. It's Cricket with a capital K. She is very, very cute. Cricket, the long-nosed pest crook, or Cricket fluffy bum crook. I'm not sure which the latest name is, but something like that. It's all right, Haley. You know, funny actually, Cricket is the only one of our pets, of which we have, as you probably know, many, that actually kept the original name that the foster, you know, the shelter gave her. All the other ones we gave different names, but Cricket just stuck for some reason. There you go. Yeah, I think so too. My youngest wanted to call her Cricket Megapaws Crook, but we Megapaws 3000, I think it was. But we shot that one down pretty quickly. All right, so test cross, right? So the problem here is what genotype does a dominant individual have? Question to the class. What genotype does a dominant individual have? Let's talk about just one gene for this case. Come on. What genotype does a dominant individual have? It's not really a trick question. It's just more than one answer. What are the two possible genotypes for a dominant individual? Yes, Sabrina's got it 100% correct. So Joshua, you're thinking about it in terms of a monohybrid or a dihybrid cross, right? So for the parent, that's correct. But Sabrina got it right for any given individual, right? Like pluck a cat off the street kind of thing. And it's expressing the dominant phenotype. It could either be heterozygous or homozygous dominant, Sabrina said, right? So if we have black cat, we actually have a black cat in the neighborhood. It's very, very cute, very friendly. Yes, exactly, Haley. So depending on what particular letters you want to use, it doesn't really matter. We'll use the same ones as we've been using. That black cat could either be homozygous dominant or it could be heterozygous. So you can't go from the phenotype to the genotype for dominant individuals, right? The only way you can do that is for recessive individuals. They have to be homozygous recessive. For dominant individuals, they could be either of these two different genotypes. So how do you figure out what it is, right? I mean, you could sequence it, right? That would be like the modern genetics way of doing it. Or if you were so inclined, you could do what's called a test cross, right? Actually, here's a much harder question to get you to really think about this, right? So let's say we have, let's move this down a little bit. You don't have to change your thing. I'm just moving it down here because that will give me the space that I need. Right, so this individual is either that or that. Okay, what genotype do you need to cross this individual to be able to see that allele? Move a little closer. Which animal, like another cat, do you need to cross this individual to be able to see that recessive allele in their offspring? Hey, really good. Thank you, everybody. That's awesome. It's got to be homozygous recessive, right? Actually, Joshua brought up an interesting point. Let's see if, the problem, Joshua, with using a heterozygote is how do you know what its genotype is? So rather than doing that, it's a lot easier to go with a homozygous recessive cat, which is, we're going to use purple cat. Right. Because if this cat had a dominant allele in its offspring, that dominant allele could potentially mask this recessive allele, right? So you want, essentially, this is like a blank slate, right? It's the white background, essentially, right? You don't want it to mask anything else. So this can only produce recessive gametes, or gametes with recessive alleles. Now, if we cross this cat over here with a cat that has this genotype, what are we going to get? So let's put it over this side. Oops, wrong color. Let's put a placeholder there. So let's say we crossed those two individuals together, right? If this individual was homozygous dominant, what offspring are you going to get from this cross? Again, you can write out the gametes. Think through it carefully, Haley. And you too, Joshua. If you cross homozygous recessive, and we're just looking at the offspring from these individuals, not subsequent crosses, just these individuals. If you put a dominant B together with a recessive B, what's the genotype of the offspring going to be? Yeah, that's right. It can only be heterozygous, nothing else. So the only genotype you're going to get if this individual is homozygous dominant is, that's a really scratchy looking cat, is going to be the dominant phenotype and the heterozygous genotype, right? So essentially, if that animal is homozygous dominant, you'll only ever get heterozygous offspring in a test cross. Did that make sense, everybody? Get my hands out of the way. Okay, good. So now, what about if we have a heterozygous individual? Now we have two different types of gametes, right? Because there are two gametes in the parent. So what different genotypes and phenotypes can we get from this individual crossed to this individual? What are we going to get now? So we're going to get either heterozygous. Yeah, you've got it all right, you two. We're either going to be heterozygous, right? I'm just going to draw it out here, the space, right? Or over here. Actually, I probably shouldn't use different colors. That's just confusing. All right, lift this up a little bit. Yeah, there we go. Or it's going to be homozygous recessive. Now, Samantha, here's a question for you. How did you get 50-50? You're 100% correct. But how did you get 50-50?
I just did the cross. Like I did a little, like the graph, and I just did it by hand. I don't know.
Oh, okay. That's cool.
Yeah.
So why do you think it's going to be 50-50? Why not like 60-40 or 30-70? Why will you get equal numbers?
Because there's a dominant in there.
And there's a
then
recessive too.
Yes.
But the key is this here. Law of segregation.
Oh.
So you're right. Don't get me wrong. You're very much right. But the reason why it's 50-50 goes back to what we did way earlier. You have equal chance of getting either one of those alleles in a gamete. And so because that's being crossed with just a, or added to just a recessive allele, you get what you said, which is 100% right. Oops, wrong color. A 1-to-1 ratio of dominant to recessive. Good stuff though. Really neat. Okay, so that in a nutshell is, oh no, a test cross. Let me just finish writing this. Like so. Yeah, super cool. I think you're all getting it. I'm really impressed. So, any questions? Let me take a quick picture of that while you have a think. You all good? All right. Okay. So let's step things up a bit. Two genes. Test cross for two genes. Now we're going to use our same applies, homozygous recessive individual here. Now, what are you going to get? Actually, let's go through it a little bit more formally. If it's homozygous dominant and you cross these two together, what are you going to get? Think back to the single gene. Yeah, that's right, Joshua. They're all going to be heterozygous. All right, if they are, or it is, actually, let's move this way. And this is going to really kind of get you thinking. What if it is heterozygous for black, homozygous dominant for fur length? So this one gene is heterozygous. Oh, don't guess, Sabrina. So think it through. We've got one gene here, which is heterozygous. That's right. So essentially, they're all going to have short fur, right? Because they're all going to be heterozygous for short fur. So we're going to have either that or that. But it's essentially the same as a single gene test cross, right? One that we just did with the black fur earlier. So you're going to have 50-50. One of the individuals is going to get the dominant allele for fur color. The other one is going to be getting the recessive allele along with this one, which is going to give you this. And so this is going to be 50-50. What about that? So this case, your homozygous dominant for fur color, your heterozygous for fur length. What are you going to get? Sabrina, let's have you thinking about this as well. Right. Awesome, Andres. Exactly. So hopefully you're seeing the pattern here, right? So now if you're homozygous dominant for black hair, all of the offspring are going to be black. So they're all going to be heterozygous. Each one will get a dominant allele, one of these two dominant alleles. So now you're going to have black cats, but you have the same deal here, single gene as heterozygous. Now you're going to get the same here, 50-50 for those two. Okay, now here's the kicker. And I'll give you a tip. Let's write this down. Think back to the F1 generation in a dihybrid cross, what gametes it can produce. Oops, wrong way around. So if you have a double heterozygote, which is essentially the same as an F1 individual in a dihybrid cross, and you cross that individual to a homozygous recessive individual, what are you going to get? Almost, Andres, but it's a little more complicated than that. So work through it. Actually, one way of doing this is, as Samantha did, make up a little, like a little Punnett square. Nope, it's not going to be 9-3-1, Joshua, because you're crossing it to a double homozygous recessive. You'd only get a 9-3-3-1 if you crossed it with another double heterozygote. This isn't a dihybrid cross, this is a test cross. So write it out. What gametes can you get from this individual? Write those out along the top. Then write the gamete that you can get from this individual on the other side of the Punnett square. Yeah, that's it, Joshua. So if those are the gametes from this individual, now just add the gametes from this individual to those, and you'll get the possible outcomes. I'll give you the first one. Yeah. So this one would be heterozygous for both. What other possibilities can you get given those gametes that Joshua has written up in the chat? Right, so you can also get, which way around did I do it? You can get heterozygous for color. That's right, Joshua. But homozygous recessive for fur length. You can get actually, that's, I don't want to confuse things. You can get homozygous recessive for fur color, heterozygous for fur length, and you can get homozygous recessive for both. And as Joshua put, the probability of getting those 4 different gametes from a double heterozygous is equal, right? You have an equal probability of getting any one of those 4. So you get a 1-to-1-to-1-to-1 ratio for the gametes because you're crossing them all to the same individual, the same recessive gametes. You're going to get a, if I was sticking with these numbers, 25, 25, 25. or 1 to 1 to 1 to 1 ratio. Ooh, my hand's going dead. Let's kind of bring this up a little closer. Oh, like so. So that's why it's important following the gametes, right? You have to work out what are the gametes that I can get here. And then you simply add those gametes to the gametes for this individual, and that gets you the different possibilities, depending on what possible gametes you can produce. All right, that's a little bit kind of more advanced. If this was a question on the test, this would be a harder question, being one of those, ooh, that's going to make me think kind of categories. But you may get a question about this sort of stuff. In fact, you'll get a question on test cross for sure, maybe a couple. And so give you another heads up too. I could also give you numbers. Let's say I could give you, we got 12 of these and 11 of these. What's the genotype of the unknown parent? Right? So you can also start it here and then work back up to this as well, right? Which is essentially how you do it as an actual experiment. So that would be a good example of one of the kind of harder type analysis questions that you will see in the exam. Any questions? This is probably something that you would benefit from going back over after class or in your revision sessions and just work through a couple of examples yourself, right? Work through each of these and see how you get these particular combinations. And again, the key is if you go slow, you follow the rules, generate the gametes, just as Joshua did, you can't get it wrong, right? Don't make a leap of faith or an assumption. Just work through the genetics and you'll always get the right answer. Almost 100% probability of that. We're going to be talking about probabilities now. All right, that's it for test plus. So now we keep talking about probabilities, right? We keep talking about ratios and what we expect, right? And really that's the domain of what we're going to be talking about next. So for the rest of the class, that took a little longer, but I think it was worth doing, particularly the test cross, because I really want to make you think about this stuff, right? To be able to actually kind of get in there and use this knowledge and information, because it's actually kind of fun, right? You might not believe me right now, but I find it super fun. Kind of, it's like simple math puzzles, right? It's kind of math that I like, not complex. And you can actually just use that and you can just work out stuff. It's really neat. It's not very often you'll get people enthusiastic about Mendelian genetics, but I actually kind of like it. Right, so probability and stats. So one reason why we teach this, or why I teach it, I guess, because I'm the only one doing it now, is that this is the whole point of Mendelian genetics, inheritance, whatever, right? It's really all about trying to figure out essentially what am I going to get, right? If I cross these two individuals with these two genotypes together, right? And then because biology is noisy, Did I get it? Right? Or not, as the case may be. Right? And because it's hard, oh, Cricks. Sorry, dog is having a nightmare. It's okay, girl. You're right. I don't know what she's dreaming about. But anyway. Where was I at? So yeah, so you can't just go by the raw numbers, essentially. Right. I got a great example of that where some of the worms that I was analyzing, I think I showed people in my lab this, they look like they're actually getting faster as they got older. So they're like, you know, kind of racy geriatrics. But that was just looking at the means. If you look at the error bars, it's all over the place. So biology is noisy, so we need to use stats to kind of cut through that noise and see what the answer is. Right? Did I actually get what I expected? Or if I got something slightly different, is it slightly different just because of noise, or is it because it's meaningfully different? Are my assumptions wrong? Right? And so this is partly a matter of Mendelian inheritance, but it's also math, right? And so really, we've got two different types of events. Oops. Man, that looks like a 3-year-old trying. So events being something that happens that you can give a probability to. So an event might be flipping a coin, right? It might be having a child. It might be stepping out into the street without looking, right? Those are all events, right, that have a certain probability of outcomes associated with them. And so which type there are depends on, I don't know how we're framing them, like what's going on at the same time. So we have independent events, right? Basically, independent events are one does not affect the other. So that sounds super similar to the law of independent assortment, right? Talking about genes. And that's essentially what law of independent assortment is. It's an independent event. What genotype you get for one gene has no effect on what genotype you get for another gene, right? They are independent events. Now, you can also have, oh man, this is going to be hard, exclusive events. Now, these aren't things that you get a special, you know, invite to. Right, a little glass of champagne. What this means, probability-wise, is if you have one outcome, you can't have the other. Well, hang on, I should put outcome there. So if you have one outcome, you can't have, oops, there we go, just managed it. So for example, if you have, and I'm not going to get into, yeah, exactly. You can't have both heads and tails, right? If you have a kid, you can't have both a boy and a girl, right? It's going to be one or the other, at least initially, right? So if you cross the road without looking, you're either going to get hit by a car or you're not, right? You can't be both, right? That's an exclusive event, right? And so generally speaking, in terms of probability, these happen in series, right? One after the other. these happen at the same time. So if you have one, it excludes the possibility of having the other, right? You can't have both of them. And as some of you may have experienced or heard, I like talking about horse racing. And this is really funny because I don't gamble, not even a little bit. I don't buy lottery tickets or anything like that. Largely because when I was a lot younger, and the UK introduced the national lottery, they said you get a 1 in 10 chance of winning like £10 or something. And each ticket was £1, right? It's like a dollar ticket. And so I bought, 10 tickets, one a week, and I didn't win £10. I was like, well, this sucks. So I never played it again. Obviously, it's a probability, right? So at that time, I don't think I had a good grasp of probability and stats. But anyway, my granddad, my dad's dad, was an avid horse racer. He was really old school, wore a flat cap. He didn't speak with a Yorkshire accent or a pirate accent, but he always had his newspaper rolled up underneath his arm so he could check off the odds and stuff like that, go down to the bookies. And he actually took me horse racing once, which was a bunch of fun. He gave me like £10 to bet on horses. I lost it all, didn't win a single penny. But horse racing is a really great way of talking about independent versus exclusive events. So that's why I like. And also I can use funny names too. So if don't put this in your SRIs because my boss reads those. And if he hears me teaching kids how to bet on horses, he might not be too impressed. Independent. There you go. All right. So from the crowd, I would like horse names. So let me pick on someone. Alfred, give me a horse name. Anything you like. No, Alfred might have gone to the restroom. Someone else, give me a horse name. Whatever you like. Could be as silly as you like, as long as it's. All right. Flicker, Buzz, and Luke. Luke, Benny, that's far too serious. I'm going to use Jubilee. There you go. Obviously, Luke is a great name for a horse, but it's not silly enough. So we've got Flicker. That's actually a good name for a horse. Buzz, as in Lightyear. By the way, don't watch Buzz Lightyear, the film. It sucks. I was super disappointed when I watched that. I thought it was going to be really cool, but it wasn't. Obviously, you can watch it and make your own decisions, but I wouldn't recommend. What? You liked it? Honestly, Haley. All right, don't worry. It's not going to affect your grade. What's the next one? Jubilee. That was a joke, by the way. Yeah, I liked the beginning of it. I thought it was really neat, but the end of it was just ridiculous. I was actually saying halfway through, because it talks about time dilation, right, which is a real serious phenomenon for space travel, right? So as you travel closer to the speed of light, you age less slowly than the people that you leave behind, right? So time dilation is a really cool thing. And I was super pumped to see it in a kid's animation, or at least an animated film. But then it just went all stupid. So anyway, what else? All right, so Flicker, we're going to have odds, right? So this is what, punters like, people who bet on the horses. We're going to have 1 in 10 chance of winning, at least according to the bookies, for Flicker, 1 in 100 for Buzz. And I'm sorry, Joshua, you are backing a nag, as they call it, 2 steps away from the glue factory, right? Yours has a 1 in 1,000 chance. Right, well, it's got a chance, right? 1 in 1,000, but it's still a chance. Obviously it has to make it to the actual finish line to win anything, but Anyway, so these are the odds, right? So if we're talking about independent events, right, we say we're talking about things happening in series, right, one after the other, right? So let's say these are all in different races, race 1, race 2, and race 3. Okay. So let's say, you know, I was my old grandparent and I gave you 10 quid to bet on a horse. And you bet on Flickr, right? So you bet on one race only, put all your money on one race, right? If you just bet on the one, you'll have a 1 in 10 chance of winning, right? Because that's the odds over here. Okay, so what about if you bet on 2 races, the first and the second race? What are your odds of winning both races, or your chosen horse, obviously, because you're not racing? Your chosen horse winning both races. Is it 1 in 10? And this is both Flickr and Buzz winning their races. Yeah, you'd have a way smaller chance. Right. So if you bet on either Flickr or Buzz, right? If you bet on Flickr, you'd have a 1 in 10 chance. If you bet on Buzz, you'd have a 1 in 100. Right, so let's move this kind of like so. Now, if you bet on both of those, right, your odds of winning both are going to be smaller than either one of these two, right? And so basically the reason being is that you're hoping for a rare event to happen twice, right? Yeah, unless you hobble the competition or you've got like Jubilee, the ancient nag, like dug out of retirement to have a race. So yeah, that, you know, we don't do that, right? That's dodgy. So now what we do is we multiply the odds of these two things together. What do you want? So instead of being 1 in 10 or 1 in 100, it's 1 in 10 times 1 in 100, right? And so now the odds of winning both is 1 in 1,000, right? What Joshua put. So another thing that my granddad used to like doing is what are called accumulator bets, right? And so this is where, you know, you hope to win a lot of money. So if you put 10 quid on Flickr and you win, you'll get £100, which is not a bad amount of money, right? You know, go out for a meal or something like that. If you win a single race bet on Buzz, you'd win £1,000, which is, that's a pretty solid chunk of cash. I would not be sad at all about winning that. If you win both of these, you bet on both of them, let's say you had 20 quid, then you'd get 1,000 times your bet, right? And so that, where the money is in terms of winnings, is what are called accumulators, right? And so what are the odds? So this is 1 in 1,000. What about if you bet on all 3 winning? So you only get a payout if all 3 of them win their respective races. What are your odds going to be for winning all 3, your horses winning all 3 races? Exactly. So now we have this probability times this probability times this probability equals, oh, running out of space. Let me bring that up over here. Right, so you've got the probability of Flicker winning, the probability of Buzz winning, and sorry, Ben, I hope I didn't offend you if you're a horse name, and the probability of Jubilee winning, right? And so to find out the probability of all three winning their respective races, right? So these are all different races. These are separate events, right? They're independent. And so the probability of that is literally 1 in a million, right? And so another way of thinking about it, which is what we talked about at the beginning, the odds of winning something are X, right? The odds of doing that twice are going to be lower, right? Whatever the odds are, it's going to be lower. Oh, so if you put, if you put £1 on each of those, you would win 3 million pounds, which is about $3 million now, but a lot of money. But also the probability of you winning that are very, very low, or is very, very low. So yeah, that's why my granddad used to pore over the paper every Saturday before the races and try and think if he could lean out like the best way of combining different horses and different races. All right, so that's independent events, right? One after the other. Okay, does that make sense, peeps? Yeah, it's like the old adage of, or the old saw of, if you win the lottery and you win a couple of £1 million or dollars or whatever, right, which would be nice, you're not going to go buy another ticket, right? Because A, you've got a load of money, so you don't need any more money potentially. But also the odds of you winning a second time are like vanishingly small. So you're literally just going to be throwing away your money. I guess why not, right? You've got £1 million to kind of like throw around. But if one were a statistician, you probably wouldn't be playing the lottery anyway. But if you were, you wouldn't be playing it after winning it. How many crickets? You can buy any number of crickets from me for that amount of money. I think she was actually $250. That was the adoption fee, or something like that. We got a discount because we were fostering her at the time. We just never gave it back. She is priceless. Yeah, you could have your own, man, imagine you could have your own farm with as many dogs as you wanted to. Wouldn't that be cool? My wife showed me like some TikTok or Instagram thing, I can't remember what, of this guy who has like a ranch or something, and he just adopts every dog that he finds, basically all the strays. And so when he goes for a walk around his property, he's got this huge train of like 25 or 30 dogs following him. So that's kind of cool. All right, so exclusive events. All right, so now these are one, not the other. So that's one way of thinking about exclusive events. The other way of thinking about it is if you were to bet on 2 horses in the same race, you potentially double your chances of winning on a horse, right? This is why I like talking about horse races. It's kind of funny, right? Because, this is part of my family history. And, I've never been to the horse races since. I've never even bet on anything since. I don't even like playing poker for pennies. Partly because I'm really bad at poker. Anyway, so if we had flicker, which was what, 1 in 10. Jubilee has retired because that was a bit too much for her, so she's going to bow out of this one. These are in the same race, right? So these are not in different races anymore. These are two horses in the same race, right? So only one of these two can win. You can't have two winners, right? Even it's like photo finish, you know, one horse is going to be ahead of the other horse at the finish line. So if you wanted to bet on both, instead of multiplying the probabilities for independent events, we add them for exclusive events, right? Because essentially, if you have one event, not another, that means you kind of double your odds, right? If you bet on both. So now, one of those winning, so you've got a 1 in 10 chance of flicker winning, right? Actually, how does this work? I'm just going to put it down here. There you go. It's easier to work in proportions rather than fractions sometimes. So you add your probabilities together. So in this case, you have a 1 in 11 chance of picking a winning horse. Essentially, one of these two may win, and the chance of one of them winning is going to be 1 in 11 or 0.11. Right? So that's a little bit harder with horse racing. Another way of looking at it is, I'm just going to take a picture here. Actually, let's get this off here. Boy or girl, right? And simply based on meiosis and sex chromosomes, you know, a male is going to produce a gamete with either a X or a Y in the sperm. So each of these has a half probability of a half. So what's the probability of getting a boy or a girl? Right, you can only have one sex in your offspring. So the sex of your child is exclusive. It's only going to be one or the other. So what's the probability when you have a given child that it's going to be a boy or a girl, right? If you were kind of betting, this is kind of bet that I would like. I don't know, I'm going to put 10 quid on it being a boy and 10 quid on it being a girl. What's my probability of winning that bet? Yeah, it's going to be 100% because if it's an exclusive event, it can only be a boy or a girl. When you flip a coin, it can only be a heads or a tail. So the probability of getting one or the other, add those together, is 1 or 100%. Right? Heads or tails is half or a half. So the probability of getting, sorry, got a text from my kid. Where was I at? Probability of getting one of those is going to be a half plus a half is 1. you'll always, whenever you flip a coin, you'll always get heads or tails, one of the two, right? You'll never get it on like the edge or something else. So that's brought us right up to and what we'll get to, Haley, what you were probably thinking about is if you have 2 children, right? Actually, let's do that very quickly. So that's exclusive boy, then girl, right? That's okay. So this is independent. This is exclusive, right? So what's the probability of getting getting a boy and then getting a girl? If they're independent events, what do you do with the probabilities when you're looking at independent events? You don't add them, you multiply them. So boy and then girl would be half times a half is a quarter. So every time you have a child, you have a 50% chance it's going to be a boy or a girl. So if you're looking at 2 children, right, and their sex is independent, so the sex you have for one child should be totally independent for the sex you have for the second child, right? Right. So those are independent events. So getting a boy and then a girl would be a half times a half, right? Or another way to look at it is I have 2 daughters. And so the probability that I was going to get 2 daughters would be a quarter, because it'd be a girl, probability of half times probability of half, which is going to be a quarter. All right, I think that's plenty enough for today. What we'll do next class is we'll look at sequence of events, right? So essentially looking at how exclusive and independent can kind of work together. So 3 children, probability to get a 3rd boy. So essentially what you're looking at there is every single time, Haley, that you have a child, the probability of getting a boy or a girl is the same. Right. So then it's not the 16th, Joshua. So actually, you know what? Let's not get into that right now because I don't have the time. People are leaving, and I'll do it next class. So keep thinking about it. We'll talk about it on Tuesday in person. Awesome. All right. Have a great day, everybody.
Professor.
Haley.
So Tuesday my power went out, so I wasn't there for lab. So I was wondering, what did I miss?
So you know that playlist on YouTube that's in the go to board picks. There's a Word document in there with a link in it. That's got the link to the playlist, and so that will, pardon me, take you to the YouTube video for the lab on Tuesday.
Okay, perfect. Yeah, because I was really worried about it. I was like, oh, this sucks. I didn't have power for a while.
No, that's okay. And that's kind of why we were doing it this way. So the video is on YouTube. The pictures of the board, such as it is, and the chat are in the, let me see. Getting you out of the way. Nah, that was such a pain in the butt. Where are we at? Here we go. Just got to make sure I give you the right information. Oh yeah. So if you go to the week that it was, right?
Okay.
All of the pictures, and there are a lot of pictures. Keep scrolling, and the chat from the Zoom are in that folder. So you can do
And
all
this
of
is
this.
due
This is due not this Saturday, the following Saturday.
next
So,
Saturday.
yeah. So
Okay.
have a look. The assignment is all online in Blackboard, as they all are. You can do it all online just with the lab handout, right? So you can use the pictures there. And yeah, I mean, it's fairly straightforward, but any hiccups you encounter, just let me know. And I can
Okay,
help you.
and everything's back to normal next week.
I hope so. Yeah, that's the plan. Should be back to normal tomorrow, to be honest, for any other classes that you have.
Well, thank you so much.
No problem. Take care.
So.
You all right, Matthew? I think